Other things cannot. Large molecules cannot easily pass through unless they are, well, fat soluble. Oh, right, like terpenes and steroids. It being a bilipid layer, I mentioned, bilipid, and I love this diagram. It depicting this bilipid layer. Oh, just, oh, welcome to
Yeah, my mom had to work all day, so
okay,
she came along.
Oh, no, the more the merrier. All right, the phospholipids, hydrophilic heads, hydrophobic tails, and then upside down. And again, I think I mentioned this, they put individual phospholipids in a test tube or a petri dish and shook them up, agitated them, and immediately they arrange themselves like this here. Okay? Because only lipids attract other lipids. Okay? Non-polar molecules, remember, this would be a non-polar molecule. So non-polar molecules will attract non-polar molecules. Okay? And that's why soap is all lipids. Okay? They put perfumes in the colonials. They used to smear the innards of dead animals on soapstone to make soap. And right, because bodily oils and well, you get the idea. Right? Lipids will attract other lipids. And again, that's a non-polar molecule. Dipolar molecules will easily be dissolved in other dipolar molecules. Not so with the polar molecules or non-polar molecules. Okay, this is what us reminiscing of that video you saw, the inside of a cell. It was always thought that before the electron microscope that all of these organelles in the cell were just floating within the cell itself. And it wasn't until the biologists were able to see exactly with the electron microscope. This is fascinating. When I was studying as an undergraduate, this had just came out. My biology instructor was so fascinated with this. The microtubercular ladder making up the skeleton of the cell. The microfilaments and microtubules. And you saw all of these microtubules being dismantled and reassembled. And again, by amino acids. Yeah, proteins. And as the name implies, microtubules are little tubes. Filaments, microfilaments, are little thread-like structures. And this would be a mitochondria, suspended or hanging out within microtubercular lattice. The nucleus and the nucleolus. Again, on a lab practical, Thursday's lab practical, a week from tomorrow. I am gonna have, maybe I'll have a cell, nucleolus, okay, listed. Okay? Or a nucleus, okay, if I put a label. Okay? And again, here, spelling, does spelling count? Hey, yeah, for the most part, but if, I'm the worst speller. But if I can differentiate, but in this case, spelling will count. There's only one or two letters
Do you
that
want us to
differentiate.
know diagrams like that for the test as well?
The what?
Do you want us to know diagrams like that for the test as well?
The exam, you should review. I'll give you hints during the review. But the lab practical, no. Just the models and the reactants that we used for the Benedict solution
I have a question.
and IUS.
About the folds, you said in the last lecture we really don't need to know much about
Yeah.
them, but it is in the study guide.
Right, yeah, and I did mention today that I will incorporate one or two
Okay, so like descriptions or pictures?
descriptions, yeah. Within my notes, okay, the heading or subheadings and the note. What I do with my exams, I cut and paste directly from my notes. I cut 4 wrong answers in and around, and then paste 1 right answer. So you have to know my notes inside and out. Okay? So again, that's the idea of the study guide. So you can rewrite my notes in actuality. This is a skeleton of my notes. So you can, so if you want, you don't have to. You can just see. But I found it would be easier for me to write when I'm seeing it, when I'm writing it, get it into my head or whatever works for you. But know my notes inside and out. Yes.
On this diagram of the nucleus, what's the difference between the chromatin and the nucleoplasm?
Oh, the chromatin, you don't see chromosomes. When do you see chromosomes? I know, we didn't go over that yet.
Is it during replication?
Right, only when a cell is getting replicated. So in this normal cell, or any normal cell, you're not going to see chromosomes. Again, I think I mentioned this before, though. When you move from place to place, what do you do with all your stuff? You package it up. So when a cell is starting to replicate, it wants to package up its DNA. So there, the chromatin will be incorporated with the DNA to make that chromosome. So again, I think I mentioned this before Monday's lecture. Most of these organelles have that bilipid, phospholipid membrane, as we saw with the plasma membrane, except for this, the membrane of the nucleus and the membrane of the nucleolus. It's not that bilipid layer. Simply because of the lot of stuff has to come in and out through those pores. The messenger RNA transcript that we saw, the ribosomes that are produced within the nucleolus or the little nucleus, they have to come out. Both from the little nucleus and then outside the nucleus itself. So again, to save energy, the passive transport type of thing. So again, they refer to that in the literature and these diagrams as the nuclear envelope. Not necessarily the nuclear membrane. Yes, it is a membrane, right? But it's very, very specific membrane. Nothing like that with any other organelle, and or the plasma membrane itself. The nucleolus. Nucleoli is one, nucleolus is one, nucleoli is many. Just like nucleus is one and nuclear, right, you get the endoplasmic reticulum, I mentioned ER is fine. There's two types. That primary structure of protein, once it gets translated by the ribosome, again, I can't emphasize this enough. Every single protein begins as a primary structure. And then gets modified into a secondary, tertiary, and or quaternary structure. And that's where the endoplasmic reticulum, the main function of, remember I refer to that primary structure as self-knitting yarn. Well, if it was self-knitting yarn, this, the endoplasmic reticulum is the way that it is being knitted itself into a scarf or a mitten. The same polypeptide chain can be modified into many, many different proteins. And that's where the epigenetics came about. My point here is that there's two types of ER or endoplasmic reticulum. If you have ribosomes adhered to the endoplasmic reticulum, that's referred to as rough ER. This would be the nucleus. Most eukaryotic cells will have the ER, rough ER, directly close to the nucleus. And it makes sense. That's where the DNA and the RNA, the RNA and the ribosome will translate the protein and then packages the self-knitting yarn within that factory of the ER. Smooth ER. Right? We see in secreting cells. Right? Our throat cells, some of our throat cells are known as goblet cells, and they will secrete mucus. Right? And those will specifically for one type of protein, that mucin, mucinological mucus. Right, so they already have their factory within. Their polypeptide chains are totally the same. Right? So again, you're going to see in some most eukaryotic cells, yes, you will see both. But again, secreting cells, smooth the Golgi body, very, very important in exocytosis. Cytosis, cyto means the cell, exo, outside, right, to exit the cell, as opposed to endocytosis. Endo within, exo is out. So within phagocytosis, to eat, phago refers to eat. So again, I'm going to be talking momentarily about these terms. Cyto, again, the cell. So this would be, they'll be used for bulk transport. Exocytosis would be mass quantities of metabolic waste products exiting the cell. Endocytosis is bringing in mass quantities of nutrients into the cell. Exiting the cell. And again, the third type of transport. Okay. Remember, passive transport, active transport, and now this third type. The Golgi body continually dismantling its membrane and producing these vesicles for exocytosis, to exiting the cell. And again, like I was saying before, the Golgi body is one of those organelles that does have a bilipid layer, similar, almost exactly the same as the plasma membrane. So when the vesicles are filled with these metabolic waste products, the vesicles are produced by the Golgi body. And when these vesicles get close to the plasma membrane, this is the outside of the plasma membrane, and this would be the intracellular. This would be extracellular on the outside within the cell. So all of these would be metabolic waste products to exit the cell. when that vesicle gets close to the plasma membrane, it will get incorporated into the phospholipids, okay, of the plasma membrane and release its metabolic waste products outside the cell. And then certain, the bloodstream will, the capillaries will take, diffuse into, and then once it's in the bloodstream, those metabolic waste products can be filtered by your liver and or your kidneys and dealt with that. The Golgi body is also responsible for producing not only the vesicles, but these lysosomes. So every single lysosome came from a Golgi body. Highly dynamic organelle. What is dynamic refer to? Ever changing. Good. Okay. So ever changing. The plasma or the membrane of the Golgi body is ever changing. Constantly. Again, the lysosomes, okay, I mentioned, produced by the Golgi body. They have the digestive enzymes, okay, and they're species specific. Some enzymes lipase, breaking down lipids, hydrochloric acid. But my main point here is that they are produced by the Golgi body. I don't care Golgi body or Golgi apparatus. Golgi is a guy's last name, so that G should be capitalized. I read a lot of his work. He started out, he meticulously mapped out the human nervous system by using a microscope, and with the other hand, he beautiful artistic work of the neurons and central nervous system and peripheral nervous. He was a neurologist. But anyways, he discovered one of those organelles, and they named organelle after mitochondria. Okay. ATP production. For now, for every one glucose molecule broken down through aerobic respiration, up to 36 ATP molecules. Again, for every one glucose molecule, up to 36 ATP molecules through aerobic respiration. If you don't have oxygen, anaerobic respiration, for every one glucose molecule, you're only getting 2 ATPs. Okay? So that's one of the advantageous aspects of the oxygen. And that's why we need oxygen so much. Okay? Again, one glucose molecule will yield up to 36. Some people's metabolic rate is higher or lower. That's the high number. But anyways, most of the ATP comes from the mitochondria. Not all. We'll see, not all, but most of it. And again, I can't emphasize this enough. You need a membrane to establish gradients. Okay? And here we're going to see two membranes. Just like in chloroplasts, we're going to see any energy needs a lot of gradients, any type of energy. And these are known as cristae. These folds, within the double membranous organelle known as the mitochondria. Cristae, and that's where most of the ATP is produced. The matrix, it's mostly water within. The outer membrane and the inner membrane. The mitochondria has its own DNA. And the chloroplast also has its own DNA. And guess what? That DNA, mitochondrial DNA and chloroplast DNA, is different from the host cell's DNA. What should that tell you? That somebody way back when in the evolutionary, one organism consumed another organism, and instead of assimilating that other organism, kept it hostage and used it for energy. And we see that in a lot of aquatic plants. The bladderwort. Right? The bladderwort, it doesn't have any roots. It's free floating. It sucks up little zooplankton, right? And uses the respiration, the carbon dioxide that little organism is producing for photosynthesis. But anyways, so that's known as the heterotrophic theory. That no, not autotrophic organisms began life. Heterotrophic organisms that consume, just like you get a new device and it's not going to work unless you put batteries in it. So I like to think that these mitochondria and chloroplasts, energy producing organelle, is a battery of the cell. And case in point, it has its own DNA.
How do you define heterotrophic here?
Heterotrophic, other energy. Right, I went over autotrophic,
The theory of heterotrophic.
right,
Oh, okay.
yeah.
I thought it was
Consuming the heterotrophic theory, the autotrophic organisms did not begin life as we know it. The heterotrophics began. They consumed. Okay,
I have a question.
yeah,
So mitochondria has its own DNA.
yeah.
Does all mitochondria have the same DNA, or they all have different DNA?
It's maternal. The DNA that you have in your mitochondria came from your mother, not from your father. And that's, we're gonna cover this a little bit. And that's the X chromosome versus the Y chromosome. The female chromosome is much, much bigger than the male chromosome.
But like, does her mitochondria have the same DNA as somebody else's, or is each?
No,
but all the mitochondria
right,
in her
yeah,
body is
yeah,
all the same?
yeah, right, yeah, that, right. But, the mitochondrial DNA, comes from your, mother, okay? Gave you the, DNA. And that's where, right, only within the family, okay? The same thing, they're similar, okay, but then, only in twins, okay, would they be exactly alike. And mitochondrial DNA in twins are exactly alike. What's different? The only thing different in twins.
Fingerprints,
Right, good. Why?
because
The fingerprint, where we get our fingerprints when the placement in our mother's womb or placenta, the fingerprints. And no, even twins have a different position around in the placenta. So the exact, everything is exact in identical twins, except for the fingerprint and the toe prints. Okay, chloroplasts. Photosynthesis, PSN. And there's two types of photosynthesis to produce. Photosynthesis, in a nutshell, you're producing carbon dioxide and water and lots of energy, photic, using the sun, photons from the sun, enzymes, lots and lots of enzymes, okay, to produce ultimately a charged sugar. This is a sugar phosphate. Okay? This is a